Inside of living cells, the regulation of gene expression can take place at any one of a number of different steps in the production of the gene products, RNA and protein. In the case of transcriptional regulation, for example, the rate at which a gene's DNA instructions are transcribed into RNA is controlled. One of the main ways this happens is through the actions of proteins called transcription factors that attach to the DNA. A simple example of this type of regulation occurs when a gene is turned off by a repressor molecule that binds to its controller region. The repressor prevents the RNA polymerase enzyme from moving along the gene and assembling the subunits needed to make messenger RNA. However, in the presence of inducer molecules, the repressor is removed and the transcription stage of gene expression can occur. In the case of post-transcriptional regulation, certain RNA binding proteins attach to the spliced, reassembled RNA transcript. Some of these gene-regulating proteins influence the RNA's stability, or determine where the RNA will be exported outside of the nucleus. With a third kind of regulation, called translational regulation, gene expression is controlled at the level of the ribosomes. In one example of this, certain gene-regulating molecules interfere with how well the large and small ribosomal subunits bind together, and so are able to control the rate of translation of messenger RNA into protein that way. A fourth place that gene expression is regulated is at the post-translational level. This generally involves changing the newly produced protein by breaking it down or making chemical alterations to it that affect how it functions in the cell.